Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Yentli Soto Albrecht, who's an MD PhD trainee whose work is sitting at an amazing intersection of genetics, neuroscience, and her own lived experience. I really encourage you to check out some of her other work and stories, but she's a carrier of the C9orf72 expansion, which is the same genetic mutation that very tragically caused her father's rapid and progressive ALS. And, we'll talk about this later in the episode. I've had, some personal tragedies in my family, and the advice that I got from the doctors is like, you have every right to let this completely crush you and take your life away from you, but you also can channel that energy in some way. And you've done that, I think, in a profound way. So we're going to share, a little bit of your story of how you've taken your research career and pivoted it to focus on this, on this area of biology. So with that, I just really appreciate you taking the time to join me.
Thank you so much, Patrick. And I really like the way that you put that notion that choosing to fight a disease that you're either predisposed to or that's really close to you head on. It's a very personal decision, and it's something that you won't really know how it impacts you on a daily basis until you try. I've actually had some people reach out to me and ask me if I can give them advice on plunging into the science of a disease that is close to their heart. And my answer for them is, you really won't know until you try. And it either adds to your life or is draining. And once you figure that out, then you'll know next steps for you.
Can you take us to the moment actually where you decided exactly this? You decided that you were going to pivot your research career and focus on C9orf72 and curing it specifically. Where were you? What were you doing? What did you feel? And how did you actually make that decision?
It was kind of a slower process that you might think. I would say in January and February of 2025, I wasn't really happy with my lot. I had just lost my dad to C9 ALS in August 2024, and I had just defended my PhD in October 2024 at the intersection of virology and mitochondrial biology. So I was really headed towards becoming an infectious disease physician scientist. But one of the things that I was really not satisfied with was my life expectancy. At that rate, I was planning on starting my lab in my 40s. And of course, as we all know, once you are done with residency as a physician scientist and you're starting your lab, it can be years before you have enough funding and the personnel to really start digging in and making those discoveries. And I was just unsatisfied with the chance of getting ALS or FTD in my 50s, which is about a 50% chance, and then in my 60s, about a 95% chance. I really wanted that time to be a scientist and delve into discoveries. And I think I balanced that with the fact that I didn't think at that time that one person would be able to make a difference in this space. I mean, scientists working on ALS and frontotemporal dementia, or FTD, the other disease that C9 causes, for decades. And it kind of feels like they haven't made a dent in it. In our families with genetic forms of these diseases, we have the same odds of dying of these diseases as we did in the generation prior. And so it was just really disheartening. But then I started realizing that one person can have an influence by just energizing other people, by lowering barriers for progress for other scientists, for raising funds or raising awareness or activating a patient population. So if you start thinking about what kind of hat you can wear given your gifts and you're flexible with what role you can play, I think one person can actually make a big difference in this space. And I'm starting to learn that. It's funny that you asked me about the exact moment. I was looking at a note that I have on my computer from February 2025, and I had just put a bunch of thoughts on a paper thinking about, like, if I wanted to accelerate a cure for C9, what would that look like? Kind of what buckets I target. And now, a year and a half later, my collaborative projects actually target all of them. So they span the buckets of biomarkers, of therapeutics, of developing research infrastructure for other scientists, and of community engagement. And I think that when you think about what it's going to take to accelerate a cure for these as of now incurable diseases, it really involves plugging holes across the entire pipeline. So not one moment, but really early 2025 is really when I started realizing that one person can do this. And as a scientist, what was really important to me too was to fall in love with the biology of what I was going to study. So I started reading about the C9 repeat expansion, and it started pulling my heart away from virology and more towards this. And that's really what allowed me to definitively say, yes, one person might be able to make a difference. Yes, my heart is here. And then from that experience, that going down this path didn't cause me pain. It gave me energy on a daily basis and took away from that cloud of my own mortality that was weighing on me. And all of that together is what allowed me to start on this path and continue on it.
Yeah, that's amazing. And I think it's actually a really good starting point for the discussion is the biology of C9 and TDP-43. How well do we understand what's going on for people who, are familiar with it? A crash course in what's going on and what we know and what we don't know, I think would be really helpful. And then I want to talk about therapeutic strategies, biomarkers, everything that comes downstream of the biology.
Sure. So the C9 repeat expansion is in an intron of a protein that has two different isomers, and we don't know exactly what the native C9 protein does, but it plays important roles in autophagy and lysosomal function. There's some papers that it helps stabilize mitochondrial complex 1 formation. So like really important roles in the cell that probably changes depending on what cell type you're setting it in. And these native proteins are expressed in neurons, but also microglia, other immune cells, and then at low levels in other cell types and across like all other organ systems from what we can tell. And there aren't really good mouse models to study C9. They're definitely worse than for SOD1 ALS, which has a genetic gene therapy treatment. And so that's probably one of the reasons why it has been understudied. So this repeat expansion is defined as anything above 30. Really 30 to 60s is intermediate. And then most repeat expansion carriers have thousands of repeats, and these are 4 Gs and 2 Cs. And so it's a hexanucleotide repeat in an intron. So it's supposed to be silent. We don't really know exactly what the protein does. And the repeat expansion, while it's supposed to be silent, it is not. So it forms 2 different types of repeat RNAs, plus and minus sense, as well as 5 different dipeptide repeat proteins, which are made during non-AUG translation. So during the integrated stress response, are preferentially translated. And they form RNA foci in the nucleus and then aggregates in the cytosol kind of throughout the life of an individual, which is a little confusing because even though the disease probably originates from this pathology, my cells are making these aggregates in RNA foci now, and I don't have disease, and I know this because I get checked annually, and so they're not really linked to disease, but they must be related to disease. And in cell and animal models, if you overexpress them enough in ways that are not native, then you can cause pathology and death. So you get the expression of these 7 products that should not be there and should not be expressed because they're in an intron. And then there's kind of a tension between the field as to whether or not there's a loss of function of the C9 proteins. In animal models, when you reduce the C9 native protein expression, it does make pathology worse. There's some thought that maybe there's not loss of function in all cell types in humans, but maybe there's loss of function in microglia or other specific subtypes where that could contribute to disease in that cell type, but not all. And then the 8th aggregate that accumulates in C9 is TDP43, which is really important alternative splicing factor. And so when TDP43 becomes dysregulated and it starts accumulating in the cytosol, you get a bunch of cryptic exons being produced that can be then discovered, targeted to develop biomarkers, but then you also get the loss of protective neuroprotective factors like FACMAN2, and so the TDP43 loss of function, and then it also starts aggregating in the cytosol, and gain of function is thought to be kind of the 8th aggregate that more closely is related to the time of onset of C9 disease, so ALS or FTD in genetic carriers. But that is still kind of correlative data, and we haven't shown this to be causative yet in people. I think something that's really interesting is that there's so many aggregates. We don't know which ones are causatively linked to disease. I think we have like some strong hypotheses, but we still need to work I think test our hypotheses in humans before we're able to really understand what that link is. And so TDP43, I just want to mention, is a really important protein, not just for C9, but it's actually becomes dysregulated in 97% of ALS, around 50% of FTD, frontotemporal dementia, about 50% of Alzheimer's, and also in traumatic brain injury, neurodegeneration. And so if we crack the code for TDP43, it might not just help a bunch of C9 patients, but also help all of these other forms of neurodegeneration as well.
Yeah, this, first of all, is an amazing overview. I think I'll just clip that section every time I need to refresher on C9 and TDP43 biology. But you led me into the next area I was curious to get your take on, which is actually the most promising therapeutic routes. It sounded like TDP43 biology in general is something that you see as there's potential for C9, but also other forms of ALS and FTD and other neurodegeneration, but there's also very targeted approaches like gene therapy, gene editing. I'm really curious to get your sense of where the focus, should be now and in the future on these different approaches.
Yeah. I think something that I'm really excited to see that's right now going into phase 2 is testing the hypothesis that if we target the TDP43 aggregates in ALS, that it's going to be disease modifying. Yep. So Vectory actually has a phase 2 trial. I think there's a couple of other companies that are soon starting or are in the progress of launching them too, where they're just targeting these TDP43 aggregates and seeing if you're able to rescue some to go back to the nucleus, but also reduce this kind of gain of function aggregate pathology. Does it ameliorate disease? I think that's something we all assume is going to work, but we haven't shown in people. And so that's pretty exciting. Other strategies are working to rectify those specific neuroprotective factors that are lost, like SAFMAN2 and UNC13A. And there's several programs, some through the Healy platform. You may have heard of CureC9, which is an organization that has one of these. They just recently discussed phase 2 data from their SAFMAN2 program. And so if there's some thought that if you rectify even just some of the factors that are misspliced and lost in TDP43 pathology, you might be able to help even some of the other factors that are misspliced. I think another approach that someone like Phil Wong has done, and I've actually talked about his work in my Search for a Self-Cure video series, explaining the science to lay people in the episode 2 on YouTube. So he is simply So we know that TDP-43 is lost from the nucleus
Yep.
in neurons that are degenerating in ALS and these other forms of neurodegeneration. And so he is simply replenishing a version of TDP-43 that is less likely to aggregate with gene therapy. And of course, for strategies like that, he is limited by gene therapy vectors, either viral or non-viral, that are able to get into enough neurons and also other glial cells that are important in disease to be able to rectify this TDP-43 function. So right now he's planning on doing so with AAV9, which is a viral vector that's been along for a while and is used in humans. But as you know, it's not going to do a great job of getting into all of the lower and upper motor neurons that we need. We're not going to be able to do multiple dosing because of the immune response. And if your cargo is too large, like if you're trying to load CRISPR into a gene therapy vehicle, then AV is not your best shot.
Right.
And so I think this kind of brings me to something that I think is going to accelerate gene therapy, not just for ALS and FTD, but for a lot of other genetic causes where we now have the tools to edit with gene editing, prime editing, CRISPR, but we don't have the way to deliver them to the cell types and organ systems that matter. Is if we really crack the code of gene therapy, we're going to be able to help a lot of people, and that's including Phil Wong, but also just about any disease where that's not in the liver, muscle, or eye, or bone.
Yeah, right. Yeah, I think that, again, this is like a super view of all the different approaches. I want to try this one more time in another area, which is biomarkers.
Yeah.
The earlier we go, the better, in theory and probably in practice. But it's, as you mentioned before, it's incompletely penetrant with respect to time, and also running prevention trials is far more challenging ethically and operationally than waiting until people are sick. But obviously, none of us want to wait until we're sick. How do you think about what we know and what we don't know about biomarkers?
Yeah. And Patrick, I would almost make biomarkers and prevention trials kind of two separate questions, because I have kind of two separate answers.
Good. Yeah, break them apart. That's absolutely right.
It's not just how early can we detect disease. And you can imagine someone like me, from my perspective, I don't know whether I'm going to get ALS or FTD. And I will tell you, I've heard experts stand up in rooms in the last year saying, we don't know if we'll ever be able to detect which disease, because we don't know where they branch off, or if they're ever going to be a point of divergence. But also tell you which decade of your life you're going to get these diseases. They could also be leveraged to screen people who are at an age risk. For example, TDP-43 pathology, which would be able to tell you, it might not tell you whether they're going to get ALS, Alzheimer's, FTD, or another form of neurodegeneration, but certainly could tell you that that person needs maybe closer follow-up with the neurologist, so that when there becomes disease-modifying treatments available, they have the best chance of protecting neurons. But then when we think about how can we run better clinical trials, we can think about using biomarkers for really smart inclusion criteria, or using them for stratifying patients into buckets, so that we can better power studies to look at differences in disease modification across different disease subgroups. Or maybe we don't have to exclude, slow progressors, because we are able to, we have good biomarkers that better power detecting progression in them, other than something like neurofilament light chain. I think there's so many ways in which better biomarkers will empower us. I think right now for ALS, what we use is neurofilament light chain. I won't even tell you what we use for FTD, because not only is NFL not as good for FTD, especially for C9 FTD, neither as sensitive or specific, but there are no C9 FTD trials, so it's like not relevant, which is a shame.
Why is that?
That's kind of wild. It's like I would, at this point, I'll either die of ALS or FTD because there's no prevention trials, or die with these diseases. But I kind of have to hope it's ALS, because otherwise I'm not eligible for a trial. I mean, that's kind of what the status quo is right now.
Why is that there's more ALS than FTD? Have you got a good answer from?
I think there's a lot of reasons. ALS has a faster progression, so it's easier to run a 6-month trial.
Yeah.
Frontotemporal dementia is a slower progressing disease. People can have it for decades. So it's harder to run a shorter trial. Also, some of the symptoms wax and wane, while ALS has very clear progression, usually with like neurofilament light chain, but also clinical scores, muscle function, and even EMGs and other corollary ancillary tests that we use. And then the only FTD treatments that are in the clinical trial pipeline right now are for progranulin. So for a different genetic cause of FTD that people thought was going to be easier to treat. And it turns out that's not true. And certainly not for C9, because C9, I just told you, the biology is very complicated. We don't even know what pathology is disease associated. And when we tried the same genetic approach to SOD1 ALS, the antisense oligonucleotide from Ionis and Biogen, that same approach failed for C9. So it is going to be harder to crack. So even for C9 FTD, there is nothing, but certainly FTD trials are a lot harder to run. So people are just assuming that we're going to have to cure ALS first and then try that same treatment in FTD. But I think with the advent of digital biomarkers, which is a different type of biomarker, or maybe digital biomarkers, biofluid biomarkers, as well as imaging biomarkers, you might be able to come up with a combination that allows you to not look at waxing and waning clinical features that have a slow decline, but look at a very well-defined pathology that's detectable through a biomarker that you'd be able to see if that curve changes on an individual basis. So that was a lot, but these are all gaps. And it turns out that the longer I'm in this space, the more I'm recognizing barriers for entities, whether they're in academia or in industry, that have the capacity and the resources to develop biomarkers, where they're also facing barriers. And for them, it may be accessing high quality patient samples, right? As simple as that. So I'm trying to figure out how I can use my role in this space, or the trifecta of being a scientist, a doctor in training, and a patient advocate, how I can leverage this towards helping these entities get access to what they need to advance biomarkers and therapeutics in this space. So that was a lot, but yeah, we're like in desperate need. And once we get good biomarkers, even just for TDP-43, but there's some like good protein, like proteomic constellations that seem to be able to predict C9 disease 5 to 10 years out. If you read Brian Trainor's more recent paper last year, it's going to take a while for those to come to clinic, but there's certainly non-TDP-43 focused biomarkers in the pipeline for C9, and that same approach is also being leveraged towards sporadic ALS and other types. So it's not going to be locked to one genetic cause.
Yeah. So tell me,
biomarkers. Do you have any follow-ups on that?
No, my follow-up is I think you've laid the land and the complexity in an amazing way with CureC9 and with your career in general. Where are you focused right now? Like, what do you see as, obviously it'll be a core bottleneck, but also one that you think you can uniquely help with? Like, if we zoom into the biomarkers or prevention trials or wherever it may be, what are you most excited about working on?
Yeah. So what I think this kind of leads itself well to where I'm learning my strong suit is in this space, and that's lowering barriers for other people to innovate. Because if you think about it, as a scientist, I could very well start my lab and dive deep into one topic, put all my eggs in that basket. But it's my survival in an incurable disease ahead of me. So I really have to think, how can I maximize my chance of winning?
Yes.
But winning being advancing a cure, whether it's me or someone else, right? Yeah. Because if there's a cure, I get to live, and that is winning. So that's lowering barriers for other people. So how can I make sure that people who are in a position of power to develop biomarkers are getting access to biofluids? How can I make sure that people who want access to patient cell lines, especially C9, are getting good quality cell lines with really good clinical and family history data? And that's why I developed the C9 biorepository, commercial biorepository for stem cells with Brain Excel. That's going to be adding around 12 cell lines a year. My dad's cells and my cells are currently being made into stem cells through that. How do we help people get access to the genetic and whole genome sequencing data that they need to be able to look at endophenotypes within disease like C9orf72 that should be less complex than sporadic ALS? Sure. We're finding is maybe more complex than we initially thought. And then how do I help people that have really promising drugs in other forms of neurodegeneration, but have promising preclinical data for ALS? How do I lower barriers for them to try their drugs in ALS? So that has been as simple as sometimes sending emails and connecting people with someone who's willing to guide them for free in this space, or recommending that they check out Experts ALS or some other platform trials, or even sometimes connecting them to my collaborator, Renzo Mancuso, if it's a drug that targets the immune response, so that he can try out this drug in a xenotransplantation model and really get to the heart of whether this drug is impacting immune cells or neurons, and kind of what the cell type, the cell target is for that therapeutic. And then I'm trying to figure out, I've signed some NDA, so I have to be careful kind of what I'm allowed to talk about. But I think in general, I'm just excited about lowering these barriers across biomarkers, across therapeutics. For me, my Search for a Self-Cure video series where I'm bringing science to patients, and the Push-Ups for ALS challenge that I did in memory of my dad, work to grow awareness about familial ALS. Those are also lowering barriers for patients to learn more about these diseases and ways in which they can engage and accelerate treatments. And the last thing that I wanted to mention is I'm probably going to be thinking about how to lower financial barriers for repositioning and resurrecting drugs in the rare disease space. So that's something that I'm talking to a couple of really smart people in this space about. And it's just about figuring out what the barriers are for people, whether that's access to samples, access to cells, access to data, access to patients, or just the landscape not being fertile ground for innovation, or for not reinventing the wheel in what is now a decade of some of the best brain penetrant compounds that we have that really should be tried across fatal neurodegenerative diseases. Why should we choose one where it might actually work across a host of diseases?
What do you see as
those are kind of all my projects, and you can see them on CureC9. And I'll add, Patrick, that these are all things that in February of 2025, I sat in an Uber and wrote on a note card. I love that. But I didn't know the experts in the field. I hadn't talked to mentors. I hadn't figured out that these were gaps. I was just like, to cure an incurable disease, it seems like these are things that I need. But what I found is that by entering the space and lowering barriers for other people to advance faster, that's how I'm maximizing my bang for my buck in this space.
Yeah, I love the strategy of try to just get as many people as you can pointed at the problem. It doesn't need to be a competitive sport, like you said. The crowd is going to do the job.
Some of them are already pointed towards it, but they're stuck. They're not advancing, or they were pointed towards it and they lost momentum because they faced a barrier that they couldn't overcome. And some of these barriers are silly. It took me 12 months to get access to my dad's cells and my cells in my lab.
Wow.
Or cells because of MTAs. And if I had that much trouble getting access to my cells to make into stem cells, could you imagine just a random person in academia? And then imagine industry. They may never get access to patient cell lines to test their therapies.
No, a lot of people would just give up if it's not personal, right?
That's right. Yeah. But I'm not going to will. If it's up to me, that's not going to happen. And then the last thing I want to mention is CureC9 is a research program that I finally put together. I didn't want to start something, but I felt like the biology of C9 is so complex, and we have really experienced some significant barriers that have caused people to lose interest in this space, like that failed antisense oligonucleotide ASO trial for C9. And so I was just thinking, I just want the smartest people that I know that are young investigators in this space that are full of innovation but haven't peaked yet. I want them thinking deeply about C9 biology outside the box a couple times a year and tell me what projects we need to accelerate. So that's the only example where I'm choosing one project and I'm accelerating it, except I'm not choosing it. I am allowing the smartest people oriented towards C9orf72 biology to choose it. And then the business advisory board, that's Indu Navar, Ed Rapp, and myself, then put our heads together and figure out what kind of solutions that project needs to move forward. And sometimes it's financial. That's why I'm fundraising $12 million for Claire Cullen's project to optimize gene therapy in the central nervous system, but also create a gene therapy atlas for academic use. But sometimes it's, if the scientific advisory board, which includes Payman, the executive director of Ionis, and Claire Cullen, the person working on CRISPR, Zach McEachin, the person that published a Cell paper on lessons learned from the C9 failed ASO trial, Chris Donnelly, and some like other fantastic scientists in this space. If they want, access to more patient samples from failed trials that they can't access otherwise, we've got two different ways we can approach it. Indu Navar, the founder and CEO of Everything ALS, and also the co-founder of CureC9, she can reach out to people at the industry partners she works with, and we can do a top-down approach of getting access to these samples, or I can leverage the patient community to write letters and to really be a pain in people's, backside to get access to these samples. So whether it's access to samples or funding or collaborators, they figure out what projects are needed, and we just find a way to move them forward.
We'll make sure to drop a link to the fundraiser on the website in the show notes. As we wrap up here, I have just one more question for you, which is really what you need help with. If people are listening and they're inspired by this, is there one or two things that you want to leave people with that they can help you with?
If you think you have a way to lower some of these barriers that I've indicated, whether it's through your network, through joining your voice with ours, through a solution that I haven't thought of, please reach out, because it's a kind of advice and help that I need. If you have a network of people that like donating to good causes and want to accelerate gene therapy for all of those diseases that have gene editing approaches but are lacking the vehicles to target those cell types and organs, seriously, anything but the liver, muscle, eye, and bone marrow, then I have a pitch deck and you could circulate it to your networks. If you are a patient or just care deeply about this and you don't have networks, but you want to donate biofluids or clinical data, there's ways to donate these that as a control and support people that you love, people in your community. You can be someone in a family impacted by genetic ALS and FTD, and you can not know your genetic status, and you can actually participate in all the same trials that I'm participating in. Those are all ALS, all FTD, dials, and everything ALS for biomarkers. And if you just want to learn, keep up to date, and figure out how you can join the fight when the time is ready, you can watch videos on Search for Self-Cure, but we can also connect on LinkedIn, because that's where I post all the updates, including about the experiment that I'm imaging at the European Synchrotron Radiation Facility today, which is why you have this great background. We're looking at metal composition in TDP TDP43 aggregates at about 15 nanometer resolution for the first time. And that's something I'm really excited about. So
very cool. Good. What a great place to be. Well, thank you. This was amazing. You're remarkable. Your mission is incredible. And I appreciate you taking the time to share it with us.
Thank you so much, Patrick. I hope you have a wonderful day. And thank you so much for having me.
You too. And thanks everybody for listening, and we'll see you next time. Thanks as always for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sanogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sanogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.